You're not just tackling a single problem. You're navigating in an array of challenges.
Most drug products have a requirement for 2 to 8 degrees of frozen storage, so that does become difficult when the typical temperature of the clean rooms is at 18 to 20 degrees centigrade.
I can think of hundreds and hundreds of container closure combinations that I've encountered just in the last year. Optimizing a container closure system requires the right people, the right environment, and the right equipment.
Shear sensitivity can impact the product's stability. Certain category of product by nature has more propensity for shear sensitivity.
Working with a light-sensitive drug product requires us to get creative in the process. It's something that we've done many times before, and we certainly don't shy away to finding the right solution for that product.
Viscous drug product filling can be very difficult. It's very important that you pay attention in order to ensure that you get very accurate and precise fill weights.
The goal is to ensure highest quality product reach the patient.
The most common temperature sensitivity that we see is maintaining the bulk drug product at 2 to 8 degrees during filling. We've also had occurrence where we need to keep the bulk drug elevated temperatures during filling. And this is typically for more viscous products, but we have the ability to do that and keep a very tight tolerance within 1 or 2 degrees centigrade. The most challenging part of filling a drug with a temperature sensitivity other than the temperature of the clean room, which is typically controlled around between 18 and 20 degrees centigrade, is to keep it cool or warm within the specific temperature range. So we will do that using a jacketed vessel with a liquid cooling system, and then we circulate the cooling fluid through the jacket to maintain the temperature within a degree or two during the filling operation. The overall typical process from formulation through aseptic filling, of course it depends on the lot size, but it's 2 to 3 days. So the formulation process could take 1 or 2 days. Filling could be 1 20-hour shift. We certainly have capabilities of doing both formulation and filling in a single day, and that's when temperature is extremely important for how long it's out of a frozen condition. There are a couple different factors you have to consider for visual inspection with temperature sensitive products. Probably the most extreme is a product that can't remain at room temperature or 2 to 8 degrees for more than a few hours. So in those cases, we will schedule the visual inspection to begin at the same time the filling operation begins. So the visual inspection will be complete, quality assurance, AQO will be complete, and the product can be at a frozen state within 2 to 3 hours of completion of fill. During 100% visual inspection of a lot, which can take 2 to 3 days, we're always very sensitive to the temperature requirements. So because of that, our recipe only allows us to remove enough product from the controlled room storage temperature. It could be 20 to 25, it could be 2 to 8, that each inspector can inspect in 1 hour. If for whatever reason they don't get finished that 1 hour's worth of inspection at the end of the hour, they have to place those units back into the controlled room storage temperature for a minimum of 1 hour, and then they can continue the inspection.
Shear force is a stress on the product when you pump a product and stress product during the high speed or pump mechanism to put the external pressure on it. And some products are sensitive to these pressure due to speed or pumping mechanism, and it may change the morphology and turns to a product degradants or like aggregation, those kind of issues. When we encounter a product that's shear sensitive, there's several ways to address it from the process perspective, right? Other than change formulation, change product itself, which is usually very upstream. But in the process development, process optimization, we look for different pumping mechanism. During filling, we could use a piston pump instead of a peristaltic pump. It may have different shear stress on the product. The other key factors, of course, the velocity. So we could adjust the pump speed to optimize in a range that has less impact on the product. The study we could design include pump the materials through different pump speed and allocate a sample from those different speed downstream and test those and see what is the cutoff point that the product can survive those speed. Similarly, with different pump mechanism, we can compare pump to pump, and sometimes we can also compare materials, what kind of tubing we use, what kind of connector we use, tubing size.
Light sensitivity can be handled in a few different ways depending on the criticality. During formulation, we can sheathe all the formulation vessels, the tubing used during aseptic filtration, and the container that we're aseptically filtering into to prevent it from the product exposure to light. During the aseptic filling process, we can do the same. We can quickly cover any trays or RTU tubs of vials filled with a sheathing as soon as it's removed from the isolator. Products that are very light sensitive can be filled into amber vials. And in the most extreme cases, we have actually changed the wavelength and the light bulbs in the room or the lenses on the light covers to accommodate whatever the product requires.
Lyophilization, most of the time, it's used when the product has inherent stability issue as a liquid form. When the product is in liquid form, it has more mobility. The molecule-to-molecule interaction or molecule-to-environment buffer or other excipients interaction is much more dynamic. When the material is in a solid state, the molecule is more confined to a limited space, so you have less chance of those degradation pathway. So lyophilization is a process to convert the material from liquid state to a solid state without impacting the product's property. In the lyophilization process, when we convert the solute in a solution to solid, is through a freeze-drying process. So freeze-drying process started with freezing the solution to a solid state, frozen. All the water becomes solid, or the solute becomes solid. And then some of the water on the surface start sublime. That sublimation process usually occurred at a lower temperature under vacuum, has less impact on the product's stability. This process could be quite long, several days sometimes. However, the outcome of this process is all the water from the initial solution will be gone, and you have the solid remaining. And those solid could form a nice looking cake, and that cake can have a much longer shelf life during storage. It started with the normal filling process to the individual vials, and these vials will be stoppered after filling. These stoppers are special stoppers suitable for lyophilization. It has a one-sided opening, and sometimes they have two-sided opening, like a 1 foot or 2 foot kind of design. And that side opening, if the stopper is partially stoppered, so it's seated well, and then put into the lyophilizer, the moisture vapor would be allowed to leave the vial. And at the end of the lyophilization cycle, those stoppers will push down and achieve the seal, and then put the cap on it, and for the final container closures. It's important to optimize the lyophilization cycle, because if the cycle parameter is not properly selected, it could have downside. If the cycle is too aggressive, meaning the cycle is too short, it may not be robust. When a cycle is not robust, either the temperature is not low enough, or the time at the primary drying or secondary drying is not long enough, you may have a final lyo cake shrink down or other undesired properties, or you may have a very high moisture level. When the cycle is too long, too conservative, you could have a much longer lyophilization cycle than required, and then economically you are making this process more costly because it will occupy the lyophilizer much longer, and the manufacturing process becomes much longer, so add the cost to the manufacturing. Optimization is to try to achieve a optimum kind of time to be the shortest lyophilization cycle, but still allow a very robust process to achieve the target product quality in terms of cake appearance, moisture level, and other stability aspect of it. Lyophilization cycle optimization, we started with the thermal characterization. One of the key property we're looking for in thermal characterization is the glass transition. Glass transition temperature is a temperature below that temperature you will see the lyo cake at the frozen state. It will maintain its physical rigidness. It will stay as a cake. But above that, it becomes glassy. You may see shrinkage. So that is the temperature we're looking for. Another key factor, the temperature we want to find out is the collapse temperature. So again, goes to the lyo cake. Once the lyo cake is formed, above certain temperature, it will collapse or shrink down, break down. But below the certain temperature, it may maintain its uniformity and intact in a nice looking cake. The cake appearance is important for two things. One is how it looks when it comes to the patient's hand, or when the cake is not uniform, it may have localized difference in degradation rate, so impacting the shelf life and the drug's quality. Annealing is needed sometimes because in the lyophilization cycle, the first step is freezing the solution down. When you freeze the solution down, a lot of times it need to go to a temperature a little below its freezing point to initiate the nucleation of crystallization. That super cooled solution, once it gets nucleated start, it is very fast process. So you may see a very variable crystal size or non-uniform solid. When you have a non-uniform solid or non-uniform crystallization, either ice or some of the solute, you may have a non-uniform lyo cake in the end. And a non-uniform lyo cake, it means variable shelf life. So it's not desired. Annealing process will help you if you heat up to a slightly elevated temperature to partially redissolve some of the content. And when you freeze down, you may achieve a better uniformity in the frozen solution. When that is achieved, it goes to the lyo cycle. The final cake would be more uniform. So that's why sometimes it's required. Following a very systematic approach using thermal characterization data to guide the cycle development, BSM can quickly develop such cycle for initial evaluation.
Viscous drug product filling can be quite difficult, and it depends upon the viscosity of the solution. Viscous products don't move well through tubing. They move very slowly. And in fact, some cases you have to apply back pressure to push the liquid through the tubing, especially when you're trying to fill a piston pump or a peristaltic pump. So that's one of the things that you need to be concerned about. Then that leads to concerns about the type of tubing that's used, because if you have a very pressurized system, that's going to cause soft silicon tubing to expand and collapse, so you can't get a really reproducible fill weight from that type of a system. Other things that you need to be concerned about are air bubbles or gas bubbles, much more significant in viscous products than they are in thin liquids, because gas bubbles can be compressed and they can expand. So when you push a liquid really hard and there's a lot of pressure built up there, that gas bubble that might be trapped in the line would compress. And after it's done compressing and the pump is done, it's a stroke, it gradually then spreads back out to where it is. So it creates a lot of dribble out of the filling needles. So there's a lot of concerns with viscous drug products. For a non-viscous drug product, typically we'd use a peristaltic pump because it's the easiest to use. It's a totally disposable fluid path that we can throw out, and when we're done with the filling operation. They can be very precise too. You can use soft silicon tubing. On the inlet side, the peristaltic pump can create enough pressure to pull the liquid into it and then push it out through the filling line. You don't have to worry about the filling lines expanding and then collapsing afterwards. But in general, we've observed here that if the viscosity gets up around 100 centipoise, then it may require some different setups. It may require a piston pump instead of a peristaltic pump. You may require some rigid tubing on the outlet side of the pump so you don't get expansion of the tubing and then that collapsing gradually, leading to variable fill volumes. We would try starting off with a peristaltic pump if the viscosity was less than 100 and see if we can get the filling rate, the number of units filled per unit time, as well as the precision of the filling volume. And that's done by running the pump at different speeds. We'd start off with a slow speed and would evaluate what type of a fill weight we get out of that. Then we would gradually increase the pump speed until you reach the point that you start to see your fill weights drop off. Because what happens is as you increase your speed and you're pumping it out, the liquid, the drug product, can't get into the pump, the peristaltic pump, fast enough to fill it up. So you'll start to see your fill weights drop off. So that will let us know what's the maximum speed at which we could run our filling pump before we're going to start sacrificing fill weight precision. So that would be the primary experiment that we'd do. If we got to a point that the flow rate, the pump speed was so slow that it wasn't worth trying to fill, we would most likely go to a piston pump. And then we'd repeat the same experiment with a piston pump, but using back pressure to push the liquid into the piston pump. A very viscous drug product would need a pressure vessel as the vessel that would be feeding the pump, and we'd be using a piston pump. We would also use very rigid tubing between the pressure vessel and the piston pump. And then we would explore various pressures to determine what's the appropriate pressure we need to push the liquid into the piston pump to fill it in a reasonable period of time. And then on the outlet side, we'd have to use a rigid tubing again, and also use the largest ID filling needle that we possibly can that would fit inside the container that's being used. Now with all of this also, you're going to want to use the largest ID and shortest length of tubing that you possibly can. All of that is going to help reduce back pressure. So that means you'd require less pressure to push the liquid into the piston pump, and then the system would generate less pressure pushing out if we keep the tubing short and the diameters as large as reasonably possible.
In a traditional fill, there are a few main sources of product holdup and loss. The redundant sterile filtration, which is oftentimes the case, the number one source of holdup. The smallest filters we have, you may only lose 4 milliliters per filter. But on some of the larger filters that are appropriate for either more viscous or larger batches, that number could be considerably higher, up to tenfold. Line losses come from tubing length, and a 1/4-inch tubing over 4 feet could very well be 25, even 40 milliliters of holdup. And once we see bubbles, we lose dose accuracy and we end the fill. most of that material will end up lost. Weight checks, destructive weight checks can account for a noteworthy amount of loss. And finally, transfers. Transfers from the container that a drug substance arrives in or during the formulation process. Transfer from one formulation vessel to another for the combination of two parts, or even just the transfer during sterile filtration from the formulation vessel to a biobag so that it can be transported to a filler for filling. Could be even 10 ml, depending on the vessel that's used. So glass vessels oftentimes have a curved base, and it can be difficult to extract all of the formulated product from the base, and that adds up quickly as well. It depends so heavily on the way we design the formulation process and the filling process. It depends heavily on the type of vessel. It depends on the diameter of the tubing. It depends on the pump seal set or the piston pump size. It depends on, in some cases, the behavior of the formulated product. If it tends to bubble, that presents an additional challenge. So it's best to do the engineering runs and make that determination empirically. Minimizing line loss, any CDMO is going to offer suggestions along the way. Filter selection's a big one. Tubing size. And again, the sensitivities, leachables, all of these things are considered by the CDMO's team of engineers that help design the process. So it's not quite as simple as saying, pick a thinner tube. But sometimes it is. We can use a smaller diameter tube and the smallest pump seal set or the smallest piston, and a smaller but effective 0.2 micron sterile filter can oftentimes get the job done. The low loss fill process at Berkshire Sterile is essentially a time pressure system, but it's designed to address all of the holdup volumes that we've discussed. We recommend a pretty short list of sterile filters. We do blow those down. When we blow down our fill lines, our formulation transfer lines, and the sterile filtration assembly, we do so at low pressure. Typically, bubble points for sterile filters are around 50 psi, so we use 5 to keep it an order of magnitude away and stay safely under the bubble point. We don't have any considerable holdup on the walls of the vessel. By blowing down the tubing and using 0.2 micron sterile filters that we have a lot of experience with, we're able to keep losses to 15, 20 grams. We've had really excellent luck and designed the system to use a stainless steel vessel that's pressurized at about 1 psi. So not a pressure that will exert enough force to cause problems for most formulations, but enough to make sure that the product is moving. But an alternative for many clients, there's a temperature sensitivity. So an alternative would be to use a jacketed glass vessel. As an example, a 6-liter jacketed vessel, we performed a fill, and the vessel holdup was 9 grams and everything else was 11. So we did come in under 20 grams for the whole process. All transfers, sterile filtration, filling and tubing, holdup, all in was about 20. So we thought that was quite successful. However, if the low loss system is able to be utilized as we envisioned it, stainless steel vessel inside a grade A space, angled downward, only 3 inches of tubing, we wholly expect to keep the entirety of loss to about 10 or 11 grams. And that's inclusive of the 7 or 8 grams of holdup in the sterile filters. We also qualified non-destructive weight checks by rolling out an analytic balance that was tuned to the environment of our laminar airflow and our grade A isolators. And in that environment, we're able to attain very steady weights, and we simply tear a vial, dispense into it, and if it's low, we can top it up so that we effectively have no underfilled units in a lot. And if it's over, we make a quick adjustment, and so that the following units are within range. The original founders selected flexible filling machines, and the nature of a flexible filling machine is it is able to adapt to different container closures. It differs from For example, an inline filler, because it may not be quite as fast, for example. But an inline filler will be set up for a single container closure, and any departure from that container closure, even the formulation of a stopper from bromobutyl to chlorobutyl, could mean you can't use that line. So a flexible filler allows us to make a lot of those changes and not decrease our efficiency or efficacy. The best course of action when bringing in a unique container closure system to any CMO is taking progressive steps of complexity, from fitting out size parts to machinability and then to engineering. And at each level, asking more in-depth questions. A fit out may just say, will this geometry work? Will the filler be able to accommodate this container closure? Whereas machinability may be able to address estimated fill speed or efficacy of plungering. And finally, the engineering is the dry run for GMP to ensure that all processes are working nominally prior to the big show. A change part or a size part is any part of the machine in a filling environment that helps mate the container closure system to the filler. So that can take the form of a plunger rod that has a suction cup to pick up a stopper and place it on top of a vial, or it can take the shape of more complex tools like the vacuum tools involved in plungering syringes. At Berkshire Sterile, the number of change parts goes up roughly orders of magnitude. So on a manual fill, we may have 2 or 3 change parts, in the rarest of occasions, perhaps 5. On our semi-automated line, 12 to 20 change parts, depending on the complexity of the fill. And finally, on our automated line, because there is not an opportunity for human intervention where humans are removed entirely from that process, there's much more conveyor movement and rotary tables, and there are size parts for every step along the way. So in that environment, there are about 200, as a rough estimate, size parts for a container closure system. So a sponsor that is bringing a custom container closure system to a CMO, or even just a container closure system that the CMO doesn't have experience with, for the outcomes to be favorable for both the CMO and the sponsor, we want to make sure that we do go through the steps of a fit out, machinability, and engineering run to gather as much data as we can on operational parameters. In some cases, we need to examine what happens on the 10,000th unit if the GMP fill expectation is 45,000 units, because some silicone, for example, may not wear in the first 5,000 units, but may begin to wear at 10,000. So there's an appropriate set of questions for each level, but running the gamut from a fit out through machinability and engineering provides plentiful evidence and data for the sponsor and the CMO to develop an effective and efficient filling program. What might appear simple on the surface, filling a solution into a container, is anything but.
Being able to adjust to different types of containers, equipment, processes is critical.
The process optimization is very important. With more understanding of the process through studies, we can better achieve the targeted product quality.
Designing an efficient and a robust filling process for a viscous product can be extremely complex.
There are many factors that we have to be aware of with a drug product. Temperature, light, shear factor are just some of the critical parameters that we have to be aware of, and then we'll be able to control for those appropriately.
Designing a robust and efficient filling program takes time, but the investment in testing and understanding parameters of filling will always result in more effective and efficient filling programs.